Cytokinins are a vital class of plant hormones that play a crucial role in regulating cell division and growth. They are especially important in plant tissue culture, where they help stimulate shoot formation and promote overall plant development. The discovery of cytokinins dates back to the 1950s, when scientists identified a substance that could promote cell division in plant tissues. This breakthrough opened new avenues in plant biology and agriculture, allowing researchers to manipulate plant growth more effectively. In plant tissue culture, cytokinins are essential for inducing shoot proliferation and preventing premature aging of tissues. They work in tandem with auxins, another group of plant hormones, to balance root and shoot development, enabling successful regeneration of whole plants from small tissue samples. There are several types of cytokinins, including Kinetin, Zeatin, and benzylaminopurin. Each type varies slightly in its chemical structure and effectiveness, but all contribute to stimulating cell division and differentiation in cultured plant tissues. Cytokinins function by binding to specific receptors in plant cells, triggering a signaling cascade that activates genes responsible for cell division and growth. This mechanism ensures precise control over plant development, making cytokinins indispensable in both research and agricultural applications.